Hi there, guys. This is Mrs. Holman, and this is part one of the introduction to the reproductive system for anatomy and physiology. If you're in my class, you have access to this PowerPoint. There are going to be links and videos throughout this PowerPoint that you can access on your own as you are processing these notes later on. But for now, we're going to get started. So today's goal for part one is that we're going to cover just an intro to reproduction, and we're going to focus on gender determination and the male reproductive system. Tomorrow, if you join us, we're going to focus on female reproduction, pregnancy, and development through early fetal and embryonic development. So let's get started. It's a good time for us to remind ourselves that back in our basic bio class, we learned that one of the characteristics of life is that all things can reproduce, which is the reason why the study of the reproductive system is so fascinating, because it's one of our basic essential characteristics of being alive. The difference between human reproduction and other animal reproduction is that we get to choose when it is that we want to reproduce. Many species have sexual selection, but we also have the ability to prevent pregnancy or to encourage pregnancy. So this is just going to introduce you to the practical guide of the reproductive system and what that means. So I just wanted to remind you guys that we're going to be culminating this unit by watching the National Geographic's version called Inside the Living Body that will take you through conception to birth. And we'll watch that video towards the end of this unit. So this is a good time to remind ourselves just about the basics of reproduction. So we can see here on the top left-hand corner of the screen, we have a paramecium dividing in half. And if we remember, that type of reproduction is asexual, and it's called binary fission. Extremely simplistic. One organism clones itself and produces two daughter species or two daughter individuals that are identically genetically identical to that original parent. In the bottom left screen, you can see that this is a common practice seen in a lot of the plant kingdom, which is called budding. So this is a particular type of plant called a hydra. It's a cnidocyte or cnidarian, which is very closely related to jellyfish, actually. And you can see that this individual will actually create an offshoot or a bud from the original parent individual. This is one type of asexual reproduction. Hydra can also go through sexual reproduction. For humans, of course, we know that that's not the case. We know that in humans, we go through sexual reproduction. And the thing that makes that exceptionally different is the fact that we spend about half of that life cycle in what's called a haploid state. So basically, the original sperm and eggs are haploid gametes, meaning that they contain half the amount of genetic information. When the sperm and the egg meet, they're going to add that genetic information together, and they're going to create a diploid zygote. So a bunch of terms swirling around in here that you're going to need to refamiliarize yourself with. Haploid means that you have half the amount of genetic information. Diploid starts with D. You can remember that that's double the amount of genetic information. We use N and 2N to represent those two values. So once fertilization has occurred, we now have a diploid zygote that's going to develop into the final mature human form, and then that's going to go through meiosis, which is the process of creating those haploid cells all over again. So in sexual reproduction, part of our goal is to get ready for that transition of creating those sex cells. And we'll be talking about those sex cells today as well. So let's move on and let's talk about benefits of sexual versus asexual reproduction. So first off, asexual reproduction has some benefits, and that is that it is fast. You don't need to spend a whole lot of energy finding a mate because there's no mate required. There's absolutely no competition, but what that means is that there's no natural selection through sexual selection either. Due to that lack of sexual selection, there's going to be much less genetic variation because each daughter is going to be essentially a genetic clone of that parent. And we now know, because of our study of evolution, that when you are less genetically varied as a species, you're going to be able to handle change in your environment a lot less. So they're going to be less adaptive to dramatic changes in their environment. Let's contrast that to sexual reproduction. So downside of sexual reproduction, much slower, and we spend a lot of energy requiring a mate. And all you have to do is look at a high school hallway for 2 seconds to see all of the energy that is involved in attracting the opposite sex. That also means that through sexual reproduction, there's going to be competition for mates. And we see this in the wild all the time. when mates will fight for territory and for breeding rights with specific other individuals. However, the benefits of this is that sexual reproduction increases genetic variation. It does that through the crossing over process in prophase 1, meaning that it'll swap around that genetic information. It also does that through random gamete combinations, meaning there are millions of possibilities, literally millions upon millions of possibilities of different sperm coming together with different eggs, and all of that combination, all of those variations mean that we're going to get a lot of genetic variation. And because of that, we're going to increase our ability for natural selection, meaning that the best traits are going to survive. And sorry, that's cut off a little bit there, but it's going to make the species stronger. And that means also, sorry, that's so cut off, that any individual or any species that goes through sexual reproduction is going to be able to adapt more quickly to a changing environment. So it's going to make them hardier whenever that environment changes. So I thought this would be a fun opportunity just to kind of start our focus of sexual reproduction, not focus on the human, but instead look a little bit into the wacky and weird world of animal reproduction. So we're going to start with octopus sex, which I think is kind of interesting. In octopus sex, the male octopus actually stores small packets of sperm, and it has one designated of its 8 legs that will take that packet of sperm and basically pass it off to the female and shove it right underneath her mantle, which is the large structure behind the eyeballs that kind of protects what would be called their head. So once it's inserted into the female's egg chamber, she'll then retrieve the sperm, fertilize her eggs, and then find a spot to deposit those eggs to give rise to new octopuses. And the really sedonic part of this is that the female will implant those baby octopus eggs in a very safe spot. She will live to guard them, protect them with her life, and as soon as they hatch, she dies. So that's pretty much the end of her life. That's normal compared to limpet sex. So limpet sex, the larva of a limpet, and a limpet is kind of like a barnacle. It stays on the surface of a rock or some sort of underwater surface. And it's kind of like a shelled organism. So they don't move very fast, and usually wherever they plant themselves, that's where they stay for the majority of their life. But limpets have the ability to actually change their gender depending on what other genders are around them. So if you think about it, it's kind of a bum luck if you are a male limpet and it just so happens that there are only other males around you. It means that your entire life is worthless because you're not going to be able to reproduce and have a baby. Because we know in biology, the level of fitness and whether or not you're successful comes from whether or not you can reproduce. So limpets will actually be able to change their sex in response to the genders of limpets around them, and it makes it so that it's always equal, nobody's left out, and everybody's able to reproduce. I think that's weird. Aphids are even weirder. So aphids are teeny little bugs that drink the sap from different plants, and aphids can either be haploid or diploid, and whether or not they're fertilized depends on the gender that they will eventually become. So females are actually produced from unfertilized egg. So if an egg the previous year does not get fertilized by a male, it will develop into a female the following year. Males will develop and hatch later in the autumn, and they will go around fertilizing as many eggs as they can. Any egg that gets fertilized becomes male, and any egg that remains unfertilized becomes a female. So the number of sets of chromosomes they have depends on determines their gender. Honeybee sex is really interesting. We know that 95% of the hive is female, and there's only 5% that are drones. Those are the males. But the drones are not those that fertilize their own hive. Drones will never fertilize anybody from their own hive because they are actually sisters and brothers of each other. So the drone's main job is to eat a lot of honey, get really, really fat and strong, and then fly away and fertilize somebody else's hive. The queen is raised from the time of the larva, and she is fed a specific type of food. It's actually called royal jelly. And that food has chemicals in it that will make her anatomically grow larger and stronger. And the queen is the only one that will actually breed and lay eggs. So about 3 days after she hatches from the honeycomb, she will actually leave the nest only one time in her life. And she secretes a pheromone that will make every drone in the Tri-County area come and find her. And in flight, they will mate multiple times. So that queen will try to mate with as many drones as she can. And she flies just fast enough that only the strongest drones can actually catch her. and they mate mid-air, and she will mate with as many of them as she can and basically collect as much sperm as she can. That's super important because when that queen gets back to the nest, she will never leave the hive again, and she will lay eggs constantly throughout the season when it's warm enough to lay eggs, and when she runs out of sperm, she dies. So pretty much as many sperms as she can get during that one flight is going to determine how long that hive is going to last with that current queen. So pretty fascinating stuff in the animal kingdom. But of course, let's come back to human reproduction, right? Now, one topic that we're going to talk a lot about, and it's important that we approach this topic with maturity and understanding, is that because this is the reproductive system, we do tend to group things in binary categories. So we'll say male and female reproduction. So it's important that on the offset, we nod and we understand that sex determination is a rather difficult thing to determine. So historically, before we had the science that we do today, the one way that we've been able to determine gender was through external genitalia. But we're learning that it's not just the physical anatomy that determines your gender. It's both physical and mental. And we're learning that the gender determination is very much a spectrum, and that there are very few cases where we are purely binary. We know from our beginning of the study of anatomy that every person can vary from every other person by at least 10%. That means that, of course, everybody is going to look a little bit different than everybody else, and that gender is never going to be purely binary. We are on a spectrum. There's been some really cool articles. This is one that I linked that shows the psychological and physical changes in gender development. It was published in Newsweek a couple of years ago, so it's a good one to look up. So if we look at how we actually determine gender, you have to kind of look at it in different levels, okay? So we're going to start with the most macro level we can get, which is, external physical appearance. So way back when, before we knew anything about anatomy and physiology, the way that you told if somebody was walking towards you and if they were male or female was purely by physical appearance. So if they culturally kept their hair longer and maybe were wearing dresses, that's girl, right? And then as we got into it, we started saying, well, that's not really fair. We need to make sure that we have a better way to determine gender. So we moved to external genitalia, where if you had the classic external genitalia of male versus female, that was the way that you were determined. The difficulty with that is that, again, there is a spectrum. So there are many people that their external genitalia is not purely binary, and they have something in between or both reproductive organs. If we look one level smaller, there's also gender determination at the chromosomal level. So we all know this from our bio class that if you have two X chromosomes, you are female. If you have an X and Y chromosome, you are male. Now, typically that seems like an easy way to determine it, except for the fact that there are a bunch of different chromosomal diseases or mutations that will kind of blur the lines of male versus female. We'll talk about those in a moment. If we go one level deeper, even one level smaller, on those chromosomes that are made up of DNA, there are DNA sequences. So we'll learn there's a specific sequence called the SRY gene that's found on the Y chromosome of males that will set into motion a domino of effects that will determine a a gender determination. So even if you have the chromosomes for one gender or the other, if you have a specific DNA sequence on a chromosome, it's going to determine gender. If we go one level deeper, so that was kind of the DNA level. If we look at hormones, this is the chemical level. So we're really at the molecular level at this point. If we look at the chemicals like estrogen and testosterone, we know that males actually produce both estrogen and testosterone. And they could have completely normal physical appearance, external genitalia, chromosomes, and genetic and DNA sequences, but maybe they produce more or less of these hormones. And because of that, those hormonal influences will put them somewhere in between the binary male-female on that spectrum. And of course, the last category is something that's not purely physical, and we are still rather limited in our understanding and knowledge of this, but that's the psychological differences between gender determination, meaning somebody might have all binary gender determination up to the psychological point, but they determine for themselves psychologically that they belong to one category or the other. And my approach to this is, I know that there's a lot of opinions out there, is that in science, we always fear the unknown. We fear things that we don't quite yet understand. And we're prejudiced against things that we don't understand. So I really hope that by learning more and by diving into the psychology of this, we can learn the science behind gender determination so that we can put a name to this, we can understand it, and then the fear and prejudice would go away. So I hope you understand all of these things. Knowing this, unfortunately, we do, since we're teaching the basics of anatomy, we are going to talk about the typical, or I use the word normal a lot. Normal does not mean anything in terms of stigma, it means statistically. So statistically, we're going to look at the normal male and female reproductive systems. So I wanted to quickly mention that gene that we talked about, the SRY gene. So the SRY gene is in charge of what we call gonadal development, so the development of the gonads. And it's found on the short end of the Y chromosome. And basically what happens is early in development, we are default female, meaning if there is no SRY present, if there's no gene present, we will automatically go through the development of ovaries, meaning that if the SRY gene is not there, we are going to default to be a female. If the SRY gene is present, it means that that gonad that is kind of bipotential, it's kind of undifferentiated, will develop into testes instead of ovaries. So really the SRY gene plays a huge role into whether or not we go to one side or the other. So if you look at the chromosome in the top left-hand corner, you can see this is the Y chromosome. It's a rather short and teeny tiny chromosome. It does not contain that many genes compared to the rest of our karyotype of chromosomes. But the one gene it does have towards the short arm is the SARI gene, and that's shown in the blue stripe. This SARI gene is on the Y chromosome, therefore we call it a sex-linked or Y-linked gene. And if you look in the bottom right-hand corner, the yellow box surrounds what we would consider a normal XY male. So that Y chromosome is an unreplicated chromosome. That SARI gene can be found there. If we look at the set of chromosomes to the left of that yellow box, we can see a XX male, which doesn't make sense because XX is typically female. But what we can see is that there was a translocation event where that SARI gene from the Y chromosome somewhere during meiosis got flip-flopped and it got put onto the X chromosome, which means that chromosomally, this is a female, it's XX. But since they still have that SARI gene present, they still have the SRY gene, they're going to develop testes and they're going to develop into a male. So this is an example of somewhere in that spectrum where even at the chromosomal level, you can't be completely certain that gender is going to be binary. If you look at the figure to the right, the XY female, that seems like a contradiction as well because XY are typically male, but because of a deletion event, that SARI gene has been removed, and therefore you can have an individual who chromosomally is XY, but is technically going to default to a female because that SRY gene is not present, and therefore they're going to develop ovaries. So as we talk about the reproductive systems of both what we call the typical male and female, we see beautiful bilateral symmetry that we see in most of the rest of our body as well. And you can see some similarities there. You can see that we both have gonads that are going to be on both the left and right side of our body. We both have a system of tubes that will lead from the gonad to a central point of convergence. For males, that would be in the urethra. For females, that would be in the uterus and eventually the cervix and vagina. So that similarity arises because technically gonads, like ovaries and testes, go through a stage in development where they're indifferent. Okay, so if we look at that top image that's kind of in the greenish teal color, this is what the reproductive systems look like early on in development. So what we see is that we see the beginning of a gonad. We see that there are two sets of ducts. So one set of duct is highlighted in teal, and it's called the Mullerian duct. That Mullerian duct, if there is no SRY present, will continue to develop and will eventually become the fallopian tubes, which is on the bottom left-hand corner shown in kind of lime green. If, however, during that indifferent stage, SRY gene is present, the Mullerian duct will actually disintegrate and go away, and the Wolffian duct will actually start to develop. The Wolffian duct, if you look in the bottom right-hand corner, would eventually become the vas deferens. And all of this is to say that early on in development, we have both male and female precursor genitalia, but it really has to do with the influence of specific genes that we have on our chromosomes that will determine the later development of those gonads and of all of those anatomical features. We also see that the tan part of each of the images here represents something called the phallus. And the phallus early on in development, depending on what genes they have, will either develop into the penis or the clitoris. So the penis and the clitoris are kind of equatable to each other, depending on whether or not we have the SRY gene. And then the genital folds, which we can see on either side of that phallus, will either become the labia in females or it will become the scrotum in males. So we can see that there's a lot of similarities between these two different genitalia, and that early on in development, it's very difficult to distinguish between the two. So this is an embryo, I should say a fetus, after 6 weeks after fertilization. It becomes it becomes a fetus after 8 weeks. So the 8-week-old embryo, you can see in the very, very bottom, that number 5, if you look at that, that's called a genital tuber. So really, it's a small little bud, and if I show you the next screen, that's kind of what it looks like in males versus females. So before about the 16 to 20 mark week of pregnancy, you really can't see much differentiation. Those two features look very much the same. After week about 16 or 20, the folds of that genitalia become more distinguished, either male or female, and it takes until about your 20-week ultrasound where they're able to determine gender using a sonogram. However, there are more genetic tests that you can do now, like the NIPS test, the non-invasive pregnancy screening, that would actually give you a chromosomal result of what your infant might be. So we're going to start with the male reproductive system. To be completely honest, the male reproductive system is a lot simpler than the female reproduction system, so that's why we're starting there. The whole system is used for 3 main purposes: intercourse, reproduction, and urination. We're going to focus on the main parts of the reproductive system: the testes, the vas deferens, the prostate, the penis, and the epididymis. So let's start here with a diagram of this system, and you can see again that bilateral symmetry. We're going to start kind of from the left side and work our way across. So we can see first on either side of the penis, we have our 2 gonads, or our testes. Wrapped around them, we have this kind of squiggly feature, which we'll talk about, that is called the epididymis. Then leading from the epididymis are these 2 tubes called the vas deferens. Those vas deferens will then fold into a gland called the seminal vesicle. That seminal vesicle will run through a second gland called the prostate gland that many of us have heard of, and then again through another gland called the Cowper's gland, which is a little less heard of. That will then run down through the opening in the penis called the urethra. Also from the urethra, we would have anything from that urinary bladder adding into that tube so that it can, again, be excreted from the body. On either side of the penis, we have this spongy-like tissue that can become filled with blood and therefore become erect, called erectile tissue. And in the very bottom, the wider part of the tip of the penis is called the glans. So those are kind of the different parts of the male reproductive system. We can see another cross section here. So this would be a sagittal section where we can see everything kind of anterior to posterior and how it lies out. We can see that the vas deferens are going to lead from the testes and then back behind the bladder. And then you can see where the urethra will start from the bladder and join kind of in this Y intersection anything that would come from the testes. They unite, pick up anything from the Cowper's gland that they need and the prostate gland that they need, and then that's when they go through the urethra, and you can see it going through the glands of the penis again. So it's a good cross shot. We're going to kind of talk about each of these parts as we come across them. So we'll start with the testes. So the testes are, again, the male gonad. They are equivalent to the ovaries. Their main job is to make sperm using meiosis and to produce testosterone. They are usually in an enclosure called the scrotum, which again develops from those folds around the genital tubule. And the main purpose of the scrotum is that they have smooth muscle that will actually contract or relax. And it's kind of brilliant because sperm need a very specific range of temperatures. So if the sperm become too cold, those muscles will contract. The scrotum will retract back up closer to the body, where the body heat from the person will actually keep those sperm a little bit warmer. The opposite happens if the person gets overheated. The scrotum muscles will actually relax, the testes will descend, and that will cool off the sperm so that they don't overheat. So again, the testicles or testes, their main job is to produce sperm and testosterone. They help with that temperature regulation depending on whether or not they will retract or descend. And you can actually live and still be fertile with 1 testes as opposed to 2. because they both produce so much. So about 5 billion sperm are produced each month, and typically males are fertile late into life, much later than females are. So testosterone itself is made in the testicles, and testosterone is that main hormone that's involved in the puberty changes, so those secondary sex characteristics that develop around ages 12, 13, 14 in males. So with those secondary sex characteristics like the physical changes, that is when males will actually produce sperm and actually get some of those more sexual urges. And when the testosterone is produced, it's going to kind of surge through the bloodstream. It's a very aggressive hormone. And this leads me to one of my favorite jokes in all of the reproductive system, which is, what is the difference between a urethra and a garden hose? Well, there's a vast difference. I'm going to hold and pause for your laughter. Anyway, so the epididymis, let's take a look at that next. So remember the epididymis, if we look at this image here, kind of lays on the outside part of the testes, and it's right before the vas deferens begins. And the epididymis' job is to collect the mature sperm as they are created in the testes. The epididymis will collect them and then propel them through the vas deferens. The vas deferens are those long tubes that are going to join up with the urethra after passing through a series of glands. And they actually have teeny tiny little cilia on the inside of them that will all beat the same direction and basically push the sperm in the correct direction. We've all heard of the word vasectomy, which if you break apart, vas comes from vas deferens, and then anything that is an ectomy is a removal of or a separation from. So like an appendectomy would be the removal of your appendix. So in the vasectomy, basically the vas deferens are either cut or cauterized. That will prevent sperm from going any further than that point in the vas deferens, and certainly not through the urethra. This is usually an office visit type of surgery. You don't have to go under general anesthesia because it is not that invasive of a surgery. And we're going to talk about that again in just one moment. So the epididymis not only collects the sperm, but they also can hold them there for weeks until those sperm are completely ready and they are mature. We'll learn the sperm have a lot of mitochondria in them so that they can swim. They need a lot of energy. They are tiny, tiny, tiny compared to the egg. And they say that enough sperm will fit on the head of a pin to repopulate the earth. So that's a lot of extra sperm that is produced. But we need that because each ejaculation event needs to flood the female reproductive system with sperm in order to increase the chances that pregnancy would occur. Sperm can also live in a hospitable environment for up to 8 days, which is kind of crazy to think about. The sperm need to have some liquid lubrication in order to reach their final destination, and that is, of course, called semen. So seminal fluid is basically sperm plus about a little less than 2 milliliters of fluid that is a combination from 3 different glands: the seminal vesicles, the prostate gland, and the Cowper's gland. The purpose of this is to nourish the sperm, because again, they're going through a lot of physical activity to swim, so they need lots of glucose, lots of nourishment, but it also helps them move by lubricating them, and it also helps to neutralize the pH of where they're headed. So the vagina, the cervix, and the uterus can sometimes have elevated or lowered pH, so the seminal fluid is meant to help kind of regulate that, so when the sperm do arrive, they won't be killed on contact. So let's talk about those 3 big glands: the seminal vesicle, the prostate, and the Cowper's gland. So the seminal vesicle's main job is to secrete the nourishing fluid. It makes up about 60% of that seminal fluid. So that's going to be the first gland. If you look at this image over to the right, the first gland that comes in contact with what is coming from the vas deferens would be the seminal vesicle. So about 60% of the seminal fluid is going to come from there. Our next stop is the prostate gland, which is one that most of us have heard of. Prostate gland is quite large, and you can see in this image it sits right underneath and kind of surrounds where the bladder dumps into the urethra. So the prostate gland makes up about 30% of the seminal fluid. Its purpose is that it creates an enzyme that will thicken the seminal fluid so that it can all gather and surround those sperm and protect them, but then it will later break down that kind of thicker substance and liquefy it so that the sperms are able to swim through the female's reproductive system. The prostate gland is also in charge of making sure that it shuts off urine during ejaculation. So since urine and seminal fluid will move through the same urethra, you definitely don't want to mix up sending out the wrong fluid. So the prostate gland will actually surround that offshoot from the bladder and enlarge to kind of pinch off that opening from the bladder during ejaculation to make sure it's only seminal fluid that is leaving. Prostate exams are done by doctors pretty commonly because of a condition called BPH, which is benign prostate hypertrophy. And this basically means that over age, or as you age, a male will slowly notice that their prostate enlarges. The reason why males typically have to go to the bathroom a little bit more, and it also slows the amount of urine that they can pee at each time. So doctors will use a rectal exam where they will actually feel through the anus and the rectum and just kind of push on the prostate to make sure that there is no malformation of the prostate. They also check during this time for prostate cancer, which is a very common type of cancer in males. So that leads us to our last gland in males, which is called the Cowper's gland. And these are teeny tiny glands. We can say here are the seminal vesicles. The orange gland is the prostate gland, and the Cowper's gland is this little appendage. It is the last gland, the last stop before you reach the full urethra. And the Cowper's gland is basically to contribute that last 10% of the seminal fluid, and it's going to secrete this clear stick of fluid, and the Cowper's gland is really there and designated to help neutralize the acidity of the urethra. So every time that urine goes to the urethra, some remnant of that urine is left there. The Cowper's gland is to make sure that the sperm won't be affected by the remnants of whatever acidic environment would be in the urethra. So we talked about vasectomies. This is 100% optional. It's 100% not a purely scientific and educational source. But if you were interested in how an outpatient vasectomy went, if you're familiar with Rhett and Link, the Good Mythical More hosts, they actually published and did a documentary on them both going through what they called a brosectomy, where they got vasectomies together. So it's a little weird, a little awkward. It doesn't show anything inappropriate, but it does let you kind of see from their point of view what that experience looks like. It's purely optional. You 100% don't need to watch that unless you're interested. So what they do during that vasectomy is, you can see this is called a no scalpel vasectomy, where they essentially go through and they just remove this passageway. They do this laparoscopically, which means they make a tiny incision in the front of the scrotum. They reach in, they divide the vas deferens. Sometimes they will cauterize those vas deferens, which will essentially make it so that sperm can't travel through anymore. Vasectomies can be reversed. And the way they do that is just by making fresh cuts on either side of the vas deferens and then stitching them together so that you can rejoin them and reverse the vasectomy. Okay. So that leads us to the last couple parts of the male reproductive system, which is the urethra. The urethra will carry both semen and urine, but they never mix. And the reason for that is due to the prostate gland and also due to sphincters, which will surround the urethra. So when a male urinates, the bladder sphincter will relax, which will let the urine pass through and go through the urethra. During ejaculation, the bladder sphincter will contract along with the prostate gland, putting pressure on that area so that only the semen can flow through to the urethra. The penis itself, at birth, the outside very end part of the penis is covered with a fold of skin called the foreskin. So we say the glans is covered by the foreskin. And an erection before ejaculation occurs because the spongy tissue on the top part of the penis basically becomes filled with blood. This is part of the sympathetic nervous system reaction of excitement. So during ejaculation, basically, those muscles will contract, the seminal fluid will be released from the vas deferens, and all of this is part of the sympathetic nervous system. So if you go down through, one of the last things we can do today is just kind of double check you're okay with this. So what I'm going to have you do is just go ahead and pause and make sure that you know where each of these are in this diagram. Okay, so we're going to go through the answers here. First one, if you look down at the tubes that are connecting the testes to the urethra are called vas deferens. The outside covering of the testes is the scrotum. The passageway for both urine and seminal fluid is urethra. The gland at the base of the bladder is the prostate. The epididymis is going to be right next to the testes that collects and matures the sperm. The bladder will hold and collect urine. The seminal vesicle will help with 60% of the seminal fluid. And then the testes is the spot where the sperm will actually be matured. Last couple things we'll talk about today is what happens for a lot of people after birth. So sometimes males are chosen by the parents to be circumcised. And basically all that means is that the foreskin that is normally found on a really young boy would be snipped so that the glands of the penis would be exposed. So there are religious reasons, and then there are medical reasons why this is done, and it all depends on personal preference. Okay, so last thing is we'll go over the timeline for male development. Basically, males can develop erections at infancy, and between ages of 11 and 14, that's when the secondary sex characteristics appear and when the beginning of sperm production starts. Technically, in their late teens, that's where the peak of sexual urges are, and they remain throughout life. There are a couple options for male contraceptive, but very few of them are approved yet. Last I checked, none of these are, where they're actually looking into ways that gels can be injected into the vas deferens. It's a more temporary situation than just getting a vasectomy. There's also a study that was done on a protein that actually affects how well sperm can swim. So with this protein, it allows sperm to really develop their tail so that as that tail twists, it projects the sperm. Without that protein, when they were genetically engineering mice to lack that protein, it would mean that the sperm would be produced and their flagellum would be a little bit messed up, which would mean that they would not be able to swim, and therefore fertility was reduced. So those are two options. The last thing I'll leave you with is just this video that goes through the different parts of the anatomy. So this is a really good review video to watch after you finish this screencast. So we're going to pause here for part 1, and tomorrow we will tackle the female reproductive system, pregnancy, and development. Hope that was helpful.